Plant breeding and biotechnology. Traditional plant breeding and biotechnology. Introduction. Plant breeding can be conveniently separated into two activities: manipulating genetic variability and plant evaluation. Historically, selection of plants was made by simply harvesting the seeds from those plants that performed best in the field. Controlled pollination of plants led to the realization that specific crosses could result in a new generation that performed better in the field than either of the parents or the progeny of subsequent generations. The expression of heterosis through hybrid vigor was observed. Traditional plant breeding. DNA is a strand of gene, much like a strand of pearls. Traditional plant breeding combines many genes at once. Traditional donor desired gene crosses with commercial variety, new variety is formed. Desired gene, new variety of desired gene is plant biotechnology. Using plant biotechnology, a single gene may be added to the strand. Desired gene transfers commercial variety, and which gives an output of new variety, which is only desired gene is transferred. Okay. Because one of the two major activities in plant breeding is manipulating genetic variability, a key prerequisite to successful plant breeding is the availability of genetic diversity. It is in this area, creating genetic diversity and manipulating genetic variability, that biotechnology, including tissue culture techniques, is having its most significant impact. In spite of the general lack of integration of most plant biotechnology and plant breeding programs, field trials of transgenic plants have recently become much more common. There are therefore reasons to believe that we are on the verge of the revolution in terms of the types and genetic makeup of our crops that has been predicted for more than a decade. More than 50 different plant species have already been genetically modified, either by vector dependent agrobacterium or vector independent biolistic microinjection and liposome methods. In almost all cases, some type of tissue culture technology has been used to recover the modified cells or tissues. In fact, tissue culture techniques have played a major role in the development of plant genetic engineering. Tissue culture will continue to play a key role in the genetic engineering process for the predicted future, especially in efficient gene transfer and transgenic plant recovery. Wide hybridization. Wide hybridization. Definition. Hybridization between individuals from different species belonging to same genus or different genera is termed as distant or wide hybridization. A critical requirement for crop improvement is the introduction of new genetic material into the cultivated lines of interest, whether via single genes through genetic engineering or multiple genes through conventional hybridization or tissue culture techniques. During fertilization in angiosperms, pollen grains must reach the stigma of the host plant, germinate and produce a pollen tube. The pollen tube must penetrate the stigma and style and reach the ovule. The discharge of sperm within the female gametophyte triggers syngamy, and the two sperm nuclei must then fuse with their respective partners. The egg nucleus and fusion nucleus then form a developing embryo and the nutritional endosperms respectively. This process can be blocked at any number of stages, resulting in a functional barrier to hybridization and the blockage of gene transfer between the two plants. Prezygotic barriers of for crossing, prezygotic, postzygotic. Prezygotic barriers, those occurring prior to fertilization, these are due to the genetic differences in different species, failure of pollen germination, slow growth of pollen tube, inability of the pollen to reach the ovary, arrest of pollen tube in style ovary and ovule. Post-zygotic barriers occurring after fertilization, hybrid in viability and weakness, leading to chromosome elimination, lethality, and embryo abortion. Hybrid sterility. Hybrid breakdown with weak or sterile individuals in F2 owing to recombination of genes, complements of the parental species. Techniques to overcome isolation barriers. Prezygotic barriers can be overcome by in vitro fertilization, protoplast fusion, embryo culture, use of growth hormones, IAA and AAA, indole acetic acid, and nicotinamide acetic acid, or I don't know what it is. So, adopting bridging species techniques. In vitro fertilization. In vitro fertilization has been used to facilitate both interspecific and intergeneric crosses to overcome physiological based self incompatibility and to produce hybrids. A wide range of plant species has been recovered through IVF via pollination of pistils and self and cross pollination of ovules. This range includes agricultural crops such as tobacco, clover, corn, rice, coal, canola, poppy, and cotton. The use of delayed pollination, distant hybridization, pollination with abortive or irradiated pollen, and physical and chemical treatment of the host ovary have been used to induce haploidy. Protoplast fusion. Protoplast fusion has often been suggested as a means of developing unique hybrid plants which cannot be produced by conventional sexual hybridization. Protoplasts can be produced from many plants, including most crop species. However, while any two plant protoplasts can be fused by chemical or physical means, production of unique somatic hybrid plants is limited by the ability to regenerate the fused product and sterility in the interspecific hybrids rather than the production of protoplasts. Perhaps the best example of the use of protoplasts to improve crop production is that of Nicotiana, where the somatic hybrid products of a chemical fusion of protoplasts have been used to modify the alkaloid and disease resistant traits of commercial tobacco cultivars. Protoplast fusion should focus on these areas. Agriculturally important traits, achieving combinations that can only be accomplished by protoplast fusion, are somatic hybrids integrated into a conventional breeding programs, and the extension of protoplast regeneration to a wider range of crop species. Embryo culture. The most common reason for the post-zygotic failure of wide hybridization is embryo abortion due to poor endosperm development. Embryo culture has been successful in overcoming this major barrier as well as solving the problems of low seed set, seed dormancy, slow seed germination, inducing embryo growth in the absence of a symbiotic partner, and the production of monoploids of barley. Interspecific and intergeneric hybrids of a number of agriculturally important crops have been successfully produced, including cotton, barley, tomato, rice, jute, hordeum, or sickle, triticum with sickle, Tripesacumex, and some brassicas. Bridging species technique. When direct cross between two species with the same or different ploidy levels are difficult to accomplish, a third species, bridge species, is used to make such crosses possible. Prezygotic barriers can be overcome by tissue culture techniques, back crossing, doubling of chromosomes, embryo rescue. Back crossing. Back crossing is a crossing of a hybrid with one of its parents or an individual genetically similar to its parents in order to achieve offspring with a genetic identity which is closer to that of the parent. It is used in horticulture, animal breeding, and in production of gene knockout organisms. Doubling of chromosomes. Artificial production of doubled haploids is important in plant breeding. Haploid cells are produced from pollen or egg cells or from other cells of the gametophyte. Then by induced or spontaneous chromosome doubling, a doubled haploid cell is produced which can be grown into a doubled haploid plant. Embryo rescue. The term embryo rescue refers to a number of in vitro techniques whose purpose is to promote the development of an immature or weak embryo into a viable plant. Embryo rescue has been widely used for producing plants from hybridizations in which failure of endosperm to properly develop causes embryo embryo abortion. Embryo rescue is one of the earliest and successful forms of in vitro culture techniques that is used to assist in the development of plant embryos that might not survive to become viable plants.